A very warm welcome to all of you guys. So today we will going to talk about the heterolactic or phosphoketolase pathway. So let's get started with the video. So this heterolactic or phosphoketolase pathway is actually a biosynthetic pathway that includes both pentose phosphate pathway and Embden-Mehra parnase pathway, that is our glycolysis, along with lactic acid fermentation. So we have already discussed about all the three, right? So that is why today we are going to talk about heterolactic or phosphoketolase pathway. Now, as I have told you that this heterolactic or phosphoketolase pathway contains pentose phosphate pathway as well as Embden-Mehra parnase pathway and lactic acid fermentation. That is the production of lactate from pyruvate, right? So the question is how the bacteria who are able to do this heterolactic or phosphoketolase pathway joins pentose phosphate pathway and Embden-Mehra parnase pathway. So the linkage between these two pathway here is a enzyme named phosphoketolase, which is the key enzyme in this pathway, and according to its name the pathway is known as phosphoketolase pathway. So as its name suggests, phosphoketolase, it have the ability to break a ketone containing compound, right? So let's see what is the heterolactic or phosphoketolase pathway. So as I have told you that this phosphoketolase pathway is essentially a combination of the initial part of the pentose phosphate pathway and the final part of the Embden-Mehra parnase pathway. And here the enzyme that play a key role is phosphoketolase. Okay, and here it is essentially a irreversible reaction and highly spontaneous because the del G0 dash here is minus 13,000 calorie per mole. Right, now you can understand how much spontaneous it is, right? So now let's directly move on to the diagram to understand the heterolactic or phosphoketolase pathway. Before that, example of heterolactic acid bacteria that have the ability to do phosphoketolase pathway are Lactobacillus aeruginosus, Leuconostoc mesenteroides, and some enteric bacteria, for example Bifidobacterium lactis. So here there is a net gain of ATP and the overall reaction is something like that. Here glucose will be converted into lactic acid along with that ethanol and CO2. Okay, so the efficiency of this reaction is half of the Embden-Mehra pathway because we are here only taking half of the Embden pathway and half of the pentose phosphate pathway. So let's directly move on to the diagram to understand the steps. So this is the diagram to understand the heterolactic phosphoketolase pathway. So it all starts with glucose. First, phosphorylation of glucose occurs so that it cannot be released from the cell membrane, right? We all know glucose is phosphorylated to trap it inside the cell. Now pentose phosphate pathway will start, and you can see that up to the synthesis of xylulose 5-phosphate all are the steps of pentose phosphate pathway. If you don't know about the pentose phosphate pathway, you may visit our playlist carbohydrate metabolism, and there you can get a separate video on pentose phosphate pathway. So here first glucose 6-phosphate will be oxidized with the help of NAD, and NAD itself will be reduced and giving us 6-phosphogluconate. Then this 6-phosphogluconate will be oxidized and decarboxylated to form ribose 5-phosphate. Then there will be a epimerase reaction and giving us xylulose 5-phosphate. Okay, now the key enzyme will come, that is phosphoketolase, and this phosphoketolase will going to break this xylulose 5-phosphate into glyceraldehyde 3-phosphate and acetyl phosphate. I hope you are already familiar with glyceraldehyde 3-phosphate and you know how to make lactate from this glyceraldehyde 3-phosphate, right? So we are not going to talk about this part because we are already know about this, that is Embden-Mehra pathway, right? But the interesting thing here to mention is the NAD, which is reduced to run the glycolysis, was recycled during the lactic acid fermentation, and we have already also talked about it in our lactic acid fermentation video. The new thing which is happening here is there will be addition of phosphate which will giving us acetyl phosphate. Okay, now this acetyl phosphate will be reduced twice to give us ethanol. Okay, and there will be also formation of acetaldehyde. So that is the thing, and interestingly this two-step reduction will going to balance this two-step oxidation. which was occurred before the cleavage. Okay, so that is the idea of our phosphoketolase pathway or heterolactic pathway. And as I have told you that there is a net gain of 1 ATP, so it is also very clear to us, right? Because you can see here we have consumed 1 ATP, right? And here we have synthesized 1 ATP. So these two things will be cancelled out, right? And this is the net gain of 1 ATP. Although here we have not considered about the NADH thing, because this 2 NADH which is oxidized here is cancelled out, right? And here also both are cancelled out. I hope you can understand, right? Because the NAD which was reduced during that step is again oxidized during that step, so we can cancel both the thing, right? And here the 2 NAD which was reduced is cancelled out by the oxidation of 2 NADH here, so there is no NAD. And here this ATP which is consumed is also cancelled out by this ATP which is produced, right? So the net gain is only this 1 ATP. Okay, so that is why I have told you that in heterolactic or phosphoketolase pathway there is net gain of 1 ATP. Okay, so now the main step here is the xylulose 5-phosphate is broken down into glyceraldehyde 3-phosphate and acetyl phosphate, right? And there is also a presence of pi, right? So this reaction is catalyzed by phosphoketolase. Remember it that this phosphoketolase also takes the help of thiamine pyrophosphate. Okay, and if we going to talk about this, the mechanism how it is done, then we came to understand that this phosphoketolase mediated step can be done by other thiamine pyrophosphate dependent enzymes, for example pyruvate oxidase or transketolase. Okay, so that is the overall idea of our heterolactic or phosphoketolase pathway. So I hope this video will be helpful for you. So thank you.